thanks. It's really nice to be here, and thanks for coming out in the rain. It's been really bad weather today, obviously. So I work with a group at Mount Sinai, and we're really interested in the intersection of deep learning, machine learning, and neuroscience, and how more understanding of the brain can lead to better machine learning algorithms, and more understanding of people as well. So this is not a very technical machine learning talk or deep learning talk, because I know more about the neuroscience. So I hope that if you know more about the deep learning stuff, then maybe this intersection will be stimulating to you. So some of the fundamental components of biological neural networks are these several components: a synaptic weight, a neurotransmitter, long-term potentiation, long-term depotentiation, and this fundamental unit that is the neuron. So the neuron has a nucleus, an axon. Along the axon is the myelin sheath. And then there's the postsynaptic cell and the presynaptic cell. And so this is, and dendrites. So these are the basic components of biological neural networks, and measuring all of the biology in there is what many neuroscientists do. Of course, there are many models that people have been developed over time to sort of approximate this using mathematics. One of the simplest approximations is the synaptic weight. So if you are good at math, then you might read that this W sub ij is the strength or the weight of the synapse. And this strength increases as you do something over and over again. So if you play piano for 10 years, the synapses will gain strength, and you'll become better at doing that thing that you do over and over again. And one of the ways that this strength is encoded is basically in the number of proteins at the synapse, or the number of connections at that site. One of the funny things that came out just earlier this year actually was that by taking electron microscope images of the synapses and axons and dendrites, what they determined is that there's a maximum of 26 distinguishable synaptic strengths. So before we didn't know whether it was from 0 to 1,000, but they've actually measured that it's, at least according to the survey, which was I think 3 electron microscope data sets, that the entire range of a synaptic weight is 26. And 26 integers is about 4.7 bits. So again, this is certainly by no means exhaustive, but what are the differences between classical machine learning or artificial intelligence and deep learning and the biological brain? Well, of course, one of the primary ones is that the human brain has many different regions, and these regions do different things for different purposes. So you have the hypothalamus, you have the substantia nigra, you have the hippocampus, you have the cortex, the neocortex, the auditory cortex, visual cortex, goes on and on and on. You can take a full 2-semester class on neuroanatomy, and you will not know everything. So, in addition, our biological brains interact with things such as supervisors, managers, and teachers, and they say things like when to stop, when to continue, when you've mastered something. And so, we don't really exist in a vacuum. There's always something that's enforcing or not enforcing some rules upon us. In addition, the brain is really very highly optimized, but what that optimization rule is highly variable and really difficult to pin down. All human brains have about 86 billion neurons, and the energy consumption is about 12 watts. And we can't just isolate the brain from the rest of the nervous system. It's actually very highly integrated with, for instance, the peripheral nervous system and the somatic nervous system and the autonomic nervous system. One might think that a convolutional neural network where you have layer 1 and layer 2 and then a convolutional piece between those is in a way a simple analogy of how different brain regions have a connection between them. So one of the things that we talk about a lot is something called the connectome. So the connectome is the network of connections in the human brain, and It's the wiring diagram of the human brain with everything you have done and everything you have the capability to do. Just see it right there or in the back. So at present, one of the most studied connectomes is the C. elegans connectome, or the flatworm connectome. And that connectome only has 302 neurons. You can download it in an Excel spreadsheet. It's 302 lines long, and it says, neuron 1 is connected to neuron 2 via axon 3. So it's, you can simply download a connectome of the C. elegans. I could send it to you if you want. But the question that often comes up when you start talking about biological brains is, how big is the human brain in bytes or gigabytes or terabytes or whatever measure you like that is a unit of bits. Here's a very schematic calculation in which it's assumed that the synapse is fully expressible in 1 byte or 8 bits. In the previous slide I said it's fully expressible in 4.7 bits, so this is actually twice too big, perhaps. So if you have 100 billion neurons and on average 10,000 synapses connect them, you get something like 909 terabytes. This is a very schematic calculation, and most people will find fault with this calculation. That's okay. Whenever I did this calculation, I could buy a terabyte hard drive for $28. So the storage for all human neurons and synapses is about $25,000. What can you get for $25,000? A new car. Would you rather have a copy of your brain or a Toyota Corolla?
Or you can pay for 3 postcards.
Yes. For a certain amount of time, right? There are so many ways you can do this calculation. Another version of it is if you have, on average, 500 inputs per neuron, the adjacency list averages out to 2 kilobits per neuron. If you assume there are 1,000 neuron subtypes, and you want to assume that the neuron can have 1,000 states. And in addition, you could do non-lossy compression on it. You can get the number down to, say, 200 to 300 terabytes. So people are doing this. They are currently decoding different pieces of the auditory and visual cortices.
So much.
The way that this was done was people were presented with some pictures inside an fMRI machine. They were lying on their back and they were shown these pictures. And then the computers reconstruct what they're looking at from this training set, which was knowledge of the fMRI data and knowledge of what they're looking at. And so you can see it's not very high resolution. fMRI is a very low resolution imaging system. It's basically 128 voxels by 128 voxels with the voxel size of about 2 to 3 millimeters. But still, it's pretty exciting. I mean, this was quite novel. In addition, people have deciphered the auditory cortex. So if you put some electrodes, many latecomers. It is only 7:15. So this one was done. Here, I'll play the clip and then I'll talk about it.
Maximum volume.
Well, it wouldn't be a meetup without tech comments, right?
structure, down, doubt.
I'll play it one more time.
Wall down, structure, down, doubt, company, pencil,
So what's happening here is there's an array of electrodes called an ECOG array that's on the auditory cortex, and a patient who's undergoing a surgery for epilepsy has this electrode array temporarily put on the surface of their brain while they're in the operating theater, and they kindly agreed to participate in this experiment while this is happening to them. And from this electrode array, you can reconstruct what they're hearing, and in some cases what they're articulating. The next couple of studies on this is they're taking a piano player and they're taking the ECOG array and they're having them play and listen to what they're playing, and then they'll have them play but not listen to what they're playing and see whether they have an unspoken or whether you can read what the pianist is feeling like they should be hearing, essentially, amongst other ways you can explain that. So really, there's a lot of auditory processing that can be read using only 64 electrodes. And one might think that this is pretty interesting, so you could potentially communicate with other people. You could just transmit information between two auditory cortices using 64 electrodes. So one of the first things that people ask when they hear about the connectome being the size that it is, and what do you do with that, is, well, can you simulate it? And so one of the many ways that you can simulate it is using something called a neuro-tissue simulator. And the details of how you construct a neural tissue simulator go on for hundreds and hundreds of pages of C++ code. But here is a loose schematic of how you can simulate neuronal tissue using a model called the Hodgkin-Huxley equations in C++. So you have a microcircuitry database, you have a database of neuronal cell types and their construction. You have a database of tissue physiology, and then you link it all together. And in the terminology of, say, computational chemistry, you integrate the equations of motion. So you chunk it time step by time step forward. So if you want to check out these couple of databases, if you go to neuromorpho.org, you can look at 34,000 different neurons. So one of the models that we think is really interesting is if you want to image not just the types of neurons, but the biophysics or the biochemistry, or what are the neurotransmitters or neuroreceptors or neurotransporters that are involved in these, you actually need a slightly higher resolution. So you need to know what are the densities of dopamine receptors, what are the densities of glutamate receptors, what are the densities of the subtypes of those receptors, because there are actually quite a number of subtypes of each of those receptors. So this is a piece of one of our spreadsheets in the lab. And so you can see that there are, a dozen adrenergic receptors, 5 dopaminergic receptors, a bunch of GABAergic receptors, and so on. It's a pretty big bunch of data. The way that we image these and the way that many other people image these is you take a ligand for these receptors, you attach, say, a gold nanoparticle to it, you put them on the cells, and wherever the gold nanoparticle sticks is where that receptor lies. But it's difficult, and many people struggle with this, including us. What would be nice is if we had a nanorobot. So this is a very science fiction-y schematic of what a nanorobot might be able to do if anyone could make these things. Trust me, no one can. So what this nanorobot does is there's a question?
Excuse me, if you'd be so kind as to speak up a bit. It's simply so noisy outside. It's getting rather hard to hear you.
I'm sorry, I'll speak up louder. As I said, this is a nanorobot. I wish I had one of these. What it does is it goes into the brain. You could inject it directly into the brain. You could inject it into the blood of an animal, and it would hopefully traverse the blood-brain barrier and then distribute itself throughout the brain in a highly nice way so that you see all the things you want to, but the odds of that happening are very, very low. But this one has power, so it's an ideal situation. So in practice, one of the many ways that people image the brain is they use an electron microscope. And so on these electron microscope studies, what you do is you take slices of tissue, you image them on an electron microscope, you take the images and put them on your computer, and you stack them up. But what you do after you've done that is you really want to know what the hell is going on. And so at this point, they leave the process to computer scientists who do things like image pattern recognition, deep learning, and so forth. And so what is illustrated in this one is, given this image stack from the electron microscope, where are all the synapses? So all the synapses are these little red pancakes. And as you can see, there are quite a lot of synapses in this little volume of 10 microns cubed. One of the things that people often ask when you get into the details of, should one or ought one to really simulate a human brain is, are the brain functions actually computable? Is there any compelling argument as to why you should or should not be able to simulate a human brain? Does it involve analog computers? Does it require quantum mechanics? Does it require whatever unknown technology we haven't discovered yet? And there's an area of mathematics called computability theory or mathematical logic, and one of the things that happens in computability theory is that, I'll just read this briefly. So the argument is that brain functions are not computable. So computability is a mathematical tool used for modeling certain systems. So that would mean that brain functions cannot be effectively modeled using computable models. This is an interesting hypothesis, but there is no scientific evidence for it. Furthermore, within the confines of current science, there is no possible way to gather solid evidence for it. The problem is that all scientific data ever gathered constitutes one large but finite set of bits. This is known as a finite set of finite precision numbers, and one of the theorems from computability theory says that any finite set of bits can be modeled computationally. So if you can disprove that, you'll be very famous. One of the other things that people often comment on is that for some reason this is not ethical, that you're meddling with the foundation of everything. And I would argue that the ethical implications are that it's ethical to not permit a tetraplegic patient or another injured person to receive a neural interface to allow them to live a more normal life, or that if you for some reason don't want to investigate connectome imaging, you're arguing that knowledge is for some reason a bad thing. It's like withholding genetic or genomic data from an oncologist or a cancer patient, which is I think it's a bad thing. When I put this talk together, I had the option of going into the details of how do you model a spiking neural network, such as our human brains, but it would be pages and pages of math. If you want to read the pages and pages of math, I recommend this paper. It's actually a really fascinating study where they look into how you can take a spiking neural network and train it to perform decisions, like play games like blackjack. It's a quite technical exposition. So, if you're interested in this stuff, what do people who work on this think is interesting? I think that one of the really fascinating things that we're currently working on, there was a DARPA meeting 2 weeks ago where they proposed in the next 3 to 4 years that there should be there should be a neural modem, and this neural modem should be able to read from 1 million neurons and stimulate 100,000 neurons, and that this should have a time frame where, year 1 it's in vitro, year 2 it's in vivo, years 3 and 4 it actually goes through the FDA approval processes for something called an investigational device exemption. So a device that is approved for research use in people. And one of the interesting things that you need to have if you want to make one of these things work is you actually have to have essentially a database of neural codes or some great understanding of how the neurons work that allows you to interpret the huge amount of data that you would get off such a device. So essentially you have a person's brain, you have the sensing and writing array, and then you have another CPU that's interpreting what all is going on. And we think that's a pretty challenging thing to work on. In addition, like the imagery construction of the synapses, taking these huge volumes of data and deciphering where are the different pieces of neurons is a really great deep learning, machine learning problem. And yeah. So I mentioned that when they decoded the auditory cortex, that it was a type of sensor called an ECOG array, which required having the skull opened and then these sensors placed on it. Presently in France, there's a new wireless version that is currently working in monkeys, and it will be approved in humans very shortly in France. That is wireless. So there are actually two antennas and no battery. One antenna is for power, one antenna is for data transmission. So you won't run out of batteries, and you can transmit and receive data wirelessly. I'd like to thank my esteemed collaborators. Regina Monaco is sitting on the side right there, and Jason Fuller is in Boston right now.
It's cool. So thanks, appreciate it. Let's open up to questions for maybe 2 or 3 minutes, and then we'll go on to the next speaker. Anybody have questions? Sure, let's go with you first.
Are there any attempts to use spiking neurons like this in more industrial settings? Like to do things that Google does with them, rather than just study the brain?
Yeah, sure. There have been companies that have been working on spiking neural network hardware forever. There's a company called Brain Corporation in San Diego, which uses a model of neuron called couch neuron. There's groups at Stanford who have spiking neural network hardware. Yeah, I mean, anybody can do a spiking neural network computer or software. Does that answer your question? Getting it to do something practical it's not working right now. It really depends on what the data is and what the study is. And the study where it counted the amount of data in the synapse, it was only 3 samples. So it varies. Sometimes it's only a very small amount. It just depends how much time and money you have to collect data. Yes?
I suppose this is purely an anatomic question, but do modern scanning electron microscopy surveys of brain reveal any information in terms of differences and expectations about neural density or volumetric density versus prior to direct imaging techniques?
Do you mean that do current imaging techniques reveal different structure than was expected, or
Do post-current imaging techniques, do our senses of mean cellular density in different regions of the brain, have they changed that much? Or are counts consistent, or have you had any surprises in terms of density across regions?
Yeah, there are surprises all the time. I mean, the synaptic thing just came out a month ago. Yeah, yes, constantly. I mean, it depends exactly what you're looking for.
All right.
And what estimate you're comparing to. I can't answer all.
Well, do you particularly specialize in the brain, so you would say the vision is we've had expectations of X neurons, and we really found that we're at 200% X or whatever in some subsystem.
I don't know any specific details that would help inform that. Yes?
What's the potential practical application of the neural modem? What would you do with it?
The primary application is interfacing with the motor cortex. So you can control arms. Basically, it's a way to control limbs. So you can control arms and legs presently you can control limbs using 6 or so electrodes. You can actually, the cochlear implant only has 4 electrodes that are working. So if you go from 4 electrodes and you can reproduce speech to 100,000 or 1 million electrodes, you can do much more in visual, auditory, and motor cortex.
So that's Darby's driver?
They don't really say what the driver is. They ask the applicants to propose an application. But most people are doing auditory, visual, and motor cortex. I think that another kind of frontier thing would be to use the stimulating electrodes to address other types of neurological disorders like Alzheimer's or Parkinson's or epilepsy, because if you can stimulate 100,000 neurons, you might be able to modulate those diseases.
One last question. Let's go there. We'll go to the next
Right there?
Yep.
I know that there's been used like these types of things, I guess, to communicate between people in the brain. How much of it is like this type of stuff more important, like reading stuff so we can understand it, and then versus how much is using this interface to actually do something, like make somebody hear something or move their arm or whatever?
It varies widely. I mean, it depends what the person wants from their neural interface. I think the communication stuff is less popular than the movement motor cortex stuff.
Great. So our next speaker is Matthew Zieler. He's the founder and CEO of Clarify. And he's giving a talk on visualizing what makes neural networks actually work.